In this video, we are going to look at the first half of the topic B2 organisation, and I've deliberately split this topic into two because it's quite a big topic. So in this video, we're just looking at the aspects of digestion and enzymes. So we kind of covered this in B1, but if we've got a group of similar cells working together, we learned a lot about cells in the topic B1, cell biology. That group of similar cells working together is known as a tissue. And several different tissues working together is known as an organ. Several organs working together is known as a system. For example, the digestive system, the reproductive system, respiratory system, circulatory system. And when those systems all work together, we have the entire organism. So we're going to journey through the digestive system now. There is a video on here where you can look at a journey through the digestive system in a more practical way, a more visual way, so that might be useful to take a look at. So all of the titles that are in blue, food actually travels in these organs, and if they're in red, then they're involved in the digestive system, but food does not enter those organs. We'll start with the mouth. We have two types of digestion in the mouth. One is mechanical digestion, and this is where the teeth break down the food into smaller pieces, increasing the surface area. There's also chemical digestion there in the form of amylase, which is a carbohydrate enzyme made in the salivary glands, and that digests starch into glucose. So starch being a large insoluble molecule, a type of carbohydrate, and then being broken down into glucose, a small soluble molecule, also a carbohydrate. Now, this isn't really that effective in humans because we don't chew our food for excessive periods of time, but in animals that chew their food a lot, amylase in the mouth can be very beneficial. The food then travels through the esophagus, and it's been bulked up with fibre. Fibre is the only nutrient that doesn't enter our blood, and it creates a nice lump of food called a bolus. And as that moves through the esophagus, the muscles above the lump of food contract, and that forces the food through the esophagus, and that process is called peristalsis. The food then enters the stomach, where there is hydrochloric acid at pH 2, and that helps to kill the bacteria present in the food, although if there are extreme quantities of bacteria such as E. coli or salmonella, then obviously it's not that effective, so food poisoning can occur. It also creates the right conditions for enzymes to work. Enzymes need optimum conditions, otherwise they denature. More about that in a moment. So the enzymes, the protease enzymes, because it's protein that gets broken down in the stomach, need acidic conditions in order to work most effectively. The stomach, because it contains all of this acid, has a mucous stomach lining which protects the stomach from the acid. Any gaps in this, if the mucus deteriorates, will cause a stomach ulcer when the acid hits the stomach wall. Now, the stomach is a giant muscle, so it also contracts in a form of mechanical digestion which churns up food, helps to mix it round, allow greater collisions of particles. There's also chemical digestion, and we must remember that it's only enzymes that can digest food. Acid can't digest food, and bile cannot digest food. Okay, so there's enzymes present in the stomach, those protease enzymes, in particular pepsin, that break the large insoluble molecules into small soluble molecules, and this is so they can be absorbed into the blood. Whilst molecules are large and insoluble, they will not be able to get into the blood, which is the transport system of the body, so they must be broken down, they must be digested so that they can be absorbed into the blood. Next we'll move on to the liver and the gallbladder. Food does not enter either of these two organs. The liver is responsible for making the bile, and it's stored in the gallbladder, and the bile is alkali. So when that food exits the stomach and goes through this small tube here called the duodenum, before it goes into the small intestine, it's joined with the bile, and that helps to neutralise that acid from the stomach so that, well, for many purposes really. One, the small intestine is not designed with the mucous lining to be able to tolerate the acidic conditions. Two, it helps to neutralise that acid so that different enzymes can work in the small intestine. For example, the amylase comes back again, and also lipase. And it also emulsifies fats. Now, this isn't digestion. This is just breaking up the fats to give them a larger surface area. So if you imagine when you eat lots of fat, it's very sticky, it sticks together. And then imagine you have a large lump of butter and you cut it with a knife and then you roll it in sand. It would be hard for that butter to stick back together again. And that's exactly what bile is doing when it emulsifies fats. It breaks globules into droplets. It increases the surface area. We call this emulsifying the fat. And because it's got a larger surface area, it means that lipase can attack it more easily when it gets into the small intestine. So now in the small intestine, we've got that bile neutralising the contents of the stomach, creating the right conditions for those enzymes, as I just said. We've also got the emulsifying of fat, increasing that surface area for lipase to digest it. In the small intestine, anything that can be digested has now been digested, okay? So now everything in that small intestine that can become small and soluble has become small and soluble and is now absorbed through the walls of the small intestine into the blood by diffusion. Now we need to be clear on these two words. Digested means taking those large molecules and making them smaller, making them soluble. And absorption is when they physically go through the wall of the small intestine, through the villi. If you remember those, give it a large surface area. We can see one on the top right there. And into the blood. And it does that by movement from a high concentration to a low concentration. When glucose moves through the wall of the small intestine, first of all, it uses the process of diffusion. And then when the concentration of glucose in the small intestine and in the blood is at an equilibrium, in other words, the same, the rest of the glucose will move across by active transport, which requires energy. Now that concept has been talked about in the B1 topic, okay? I'm just doing a bit of a recap there. So the villi have a large surface area, again that was in B1, to allow maximum diffusion of these small soluble substances into the blood, into that transport network of the body. Any undigested food moves on to the large intestine, again by peristalsis. Any movement through tubes is done by peristalsis. And vitamins and minerals don't need to be digested because they're already small and soluble. So they don't need to be broken down. They can be absorbed straight into the blood without any form of digestion. Our last stop then is the large intestine, where any water remaining in the large intestine can be absorbed into the blood. Okay, and then waste is removed through the rectum and then the anus. So let's look at our summary then. We must remember that enzymes are biological catalysts made of protein. Okay, so they are these long chains of protein that have been wound up on themselves, cross bonds between them. Okay, and it's the extreme temperature and the extreme, extreme heat, should I say, not extreme cold, just extreme heat, and the extreme pH that will cause some of those cross bonds to break and therefore unravel that protein and change the shape of the active site. If you don't get that bit, don't worry about it too much, it's triple content. We come up against it again in paper 2. So we've got 7 different nutrients, okay? We've got the fibre, which is not digested. It just builds up the food so that it can travel through the digestive system. We've got water, which just helps the whole process. Vitamins and minerals, which didn't need to be digested. So it just leaves us with our last 3 nutrient groups, the carbohydrates, the proteins and the fats. Okay, so we're looking at the 3 categories of enzymes which will break down these 3 food nutrient groups. Start with the carbohydrates. An example is amylase. It's made in the salivary gland. Again, we said it's not massively effective in the mouth. It then denatures when it gets to the stomach because of the acidic conditions, and then it's produced again by the small intestine and the pancreas for use in the small intestine. Remember, food does not enter the pancreas. And what that amylase does is it takes the large insoluble molecule, which is starch, and digests it, breaks it down into the small soluble molecule, which is glucose. And that's going to be used for respiration. Then we've got our protease, slightly more complicated, this one. We've got two examples. We've got pepsin, which works in the stomach. It thrives in acidic conditions. And then we've got trypsin, which works in the small intestine, and that works best in more alkali conditions. And if we are looking at doing the triple science exam, then we actually know there's an in-between stage here. So we've got protein. Now the pepsin turns that protein into peptides, and then the trypsin takes the peptides and breaks those down into amino acids. Okay, so it's like a two-stage process. Part of that protein being digested in the stomach, part of that protein being digested in the small intestine. Okay, again, the amino acids are the small soluble molecules. And if you notice here, if in doubt, all of those enzymes are made in the small intestine, all are made in the pancreas. We've got the addition of the protease in the stomach, and we've got the carbohydrates in the salivary gland. And then we move on to lipase. They do not want us to know any specific example of a lipase enzyme, but we must remember that there are two products of the digestion of fat, okay, which can be called lipids, and that is fatty acids and glycerol. Okay, and that will take place in the small intestine with that lipase being made in the small intestine and the pancreas. So once that fat has been emulsified by bile, larger surface area, these droplets of fat, then lipase can do its job in the small intestine. Okay, and then we just got our summary there again. I'm going to say it one more time. Enzymes digest large insoluble molecules into small soluble molecules so they can be absorbed through the wall of the small intestine, and soluble means they can dissolve, so they have to be dissolved in a liquid in order to travel through the villi of the small intestine and into the blood. Okay, so enzymes are specific. Only one type of substrate, so that could be the carbohydrate, the protein or the fat, will fit into the active site. And we call that the lock and key model, the fact that there is a specific complementary shape of the enzyme and the substrate. Okay, and they work best in their optimum conditions. Okay, so having the right pH and having the right temperature. At low temperatures, the particles just don't have much energy, so we don't see many collisions, as we learn about in chemistry. And in extreme heat, denaturing can occur. And denaturing is where I said you've got this protein. Okay, so the enzyme is a protein, and it's kind of folded up on itself, raveled together with these cross links between them. And when that heat gets too hot, or the pH becomes too extreme, those cross links break, those bonds break, the protein unravels a bit, and it changes the shape of the active site. So if the active site changes shape, then the lock and key no longer fit together. So therefore, it will not work as an enzyme anymore. Okay, enzymes aren't used up so long as they don't get denatured. They're not used up, and they can be reused. But once they're moving through the digestive system, the pH changes and they denature, which isn't always a problem. Okay, so once it's done its job, it doesn't matter if it denatures after that. We do get some denaturing of the amylase from the salivary gland in the stomach before it gets to the small intestine. So we make fresh amylase for the small intestine. So we've got this phrase activation energy, which I'm sure we've heard in chemistry. And this is saying that when particles bounce around and collide off each other, sometimes they collide with enough energy to react, sometimes they don't have enough energy and they just bounce off each other and no reaction occurs. Now, when they bounce off each other and they do have enough energy to react, we call this the activation energy. And what enzymes do, because they're catalysts, is they reduce the activation energy, which means that particles that would have in the past just bounced off each other and not reacted will now react to make new products. This page really just highlights that there are two kinds of enzymes. The ones that break down molecules, which are used in digestion, are called catabolic enzymes. But also there are enzymes which join particles together. For example, when plants photosynthesize and they make glucose, and that glucose is joined together as a storage of starch, that's done by an anabolic enzyme. We don't really need to know all of that detail, but it's just interesting to know that they don't just break down particles, they can build them up as well. Bit more here on denaturing. So we know that at low temperatures we see greater movement, greater energy, greater collisions, therefore a faster rate of reaction. We then reach the optimum temperature where we've got maximum activity from the enzyme, and then we get denaturing, where that protein, the bonds break, it changes shape, and therefore the substrate will no longer fit. And we can see that shape change here by that extreme heat. It's irreversible, okay? We can't do anything to the enzyme to turn it back into the shape it was originally. Hopefully this graph looks quite familiar. We're looking at the two enzymes here, pepsin and trypsin. Pepsin working in the stomach at that lower pH optimum, and trypsin working in the small intestine at a much higher pH. We can also see, if we were really analysing this graph, we can also see that trypsin is more effective than pepsin because it has a higher peak. Also works over a wider range of pH. Okay, so pepsin taking the proteins, breaking them down into peptides, and then trypsin taking those peptides and breaking them down into amino acids. And it's the hydrochloric acid that creates the right conditions for pepsin to work, and it's the bile that creates the right conditions for trypsin to work. Then we came across a required practical. This one was the really tricky one. It was tricky because it's hard practical to get results for. So every table in the room had some amylase at a different pH using a buffer solution. So we put this buffer solution in and it adjusts the pH of the amylase. And then we mixed a fixed volume of amylase with a fixed volume of starch. And that amylase was working like scissors to chop down the starch. And when that starch was in that solution, we tested it every few seconds to see had all the starch been chopped up and broken down. And we did that using iodine solution, which stays orange when the starch is all gone, and if there is starch present it turns blue-black. So what we did is we had that starch, we added our particular amylase with whatever pH it was, we tested that pH using some universal indicator paper, and then every few seconds test it until we get no colour change. When the iodine solution no longer changes colour, we know all of the starch has been broken down by that particular amylase solution at whatever pH it was. And if we just take the results with how long it takes for that digestion of starch to occur, we can see that a short time here at pH 7 means that pH 7 is the optimum pH. Now we may get a graph like this where we've got time here and we say, okay, quick time means fast rate of reaction, pH 7, or we may have to convert it into a rate of reaction graph, which I've actually just taken this graph and flipped it like this, and it gives me more of that pattern that we saw back here where we've got the peak at the top of the highest rate of reaction. Now if we needed to calculate that in the exam, we can do rate of reaction is 1000 divided by time. So take 1000, divide it by say 10 seconds, and we can work out the rate of reaction, then plot that as a graph. That's something I see coming up at some point in time. Okay, and then again we can see that pH 7, fastest rate of reaction, has the peak there on the graph. And then we came to our next, this is our last page, we came to our next required practical, which was the food testing. And this really is likely to be our 6 marker, or a shorter one if it's just looking at one of the tests here, one of the chemicals. And it really is a case of not just saying that there will be a colour change, but you need to know the starting colour, the ending positive test colour, the name of the chemical and what it is testing. So we'll start at the top. We've got the Benedict solution testing for sugar. Now we can't say testing for carbohydrate because both of these are carbohydrates. So we've got to say specifically testing for sugar. And the Benedict solution begins this blue colour, and I remember this like traffic lights, okay? Little bit of sugar present, green, little bit more, we've got the orange, and if there's a lot of sugar present, we get the red. Now, one thing that's not written on here that we must write down is that Benedict solution is heat activated. So it must be warm in order to react with the sugar in the food. Okay, if you just leave it there, you could leave it all day and you won't get any reaction. So it's heat activated, and then we've got that traffic light colour change showing us how much sugar is present in the food. The starch test I think people are quite confident with. We use iodine solution quite often. We can see here, this is a bit like the required practical we just talked about, where we test every few seconds. We see there's still lots of starch present here, and then as time goes on, less and less starch present. It's all been chopped down by that amylase into glucose, and we can see we've got no colour change here. So that gives us an indication of how quickly the amylase broke down the starch solution. And if we have got starch present in a food, for example potatoes, then we will see that blue-black colour change, and if there's no starch, the iodine solution will remain orange. And the last one here is the protein test, the biuret solution, and this is again, it starts off blue and it's purple for protein. So we've got a bit of alliteration there, purple for protein. And it's quite good to put some of the biuret solution in a tube and just have it as a comparison, because it's quite a subtle colour change. So if we have one that we can compare to, we can see, oh yeah, it has actually changed to purple, therefore there is protein present in that food. So if we had a 6 marker that said, how do we test for different nutrients in food or something like that, then they're suggesting we talk about each. So we could separate it with our subheadings. Which chemical do I use? Which colour change am I expecting for a positive result? Okay, so that is half of B2, and we will come back to the other half of B2 later. But we've now covered everything for digestion enzymes. I hope you found that useful. Don't forget to hit subscribe. Thank you.